Hi everyone, it's Dr. B here, and this is Physical Principles of Quantum Biology, Chapter 4, Photosynthesis and Open Quantum System Dynamics. This is from my new book with my wife Brandy, Physical Principles of Quantum Biology. You can download your copy from the archive for free. And today, as I mentioned on the opening slide, we'll be looking at Chapter 4 from the table of contents, Photosynthesis and Open Quantum System Dynamics. So let's begin. What are the essentials and what are the open questions surrounding photosynthesis? In this lecture, we'll be exploring how quantum electrodynamics govern photosynthetic energy transfer through vibronic coherences and environmental dissipation. These kind of effects basically lay the foundations for all of life as we know it. Oxygenic photosynthesis converts sunlight to chemical energy, splitting water molecules to produce the oxygen that we breathe. While photosynthetic structures are well characterized, everyone studies them in their introductory biology courses, as it stands, predicting the functional outcomes of these systems from the microscopic quantum dynamics remains very challenging. And so we'll really be considering the question of how do light-matter interactions and electron transport physics connect to macroscopic efficiencies and regulation mechanisms. Photosynthesis begins with an exciton transfer paradigm. Just as we considered electron transfer in our look at respiration, exciton transfer is the transfer of an excited electronic state. The electron itself doesn't need to transfer per se. It's the excitation of a molecule, such as a beta-carotene molecule or a bacteria chlorophyll molecule, and the excitation can be passed from one molecule to the next. So during photosynthesis, we have the basic premise that light is absorbed in the first step, absorbed in fact by multiple chlorophylls that capture photons collectively. The exciton is formed after the photon is captured, creating an excitation. It can delocalize across many chlorophyll pigments, often bacterial chlorophyll. These are chlorophyll in bacteria because bacteria are kind of classic model organisms to study in our biology labs. Now, once the exciton forms, it needs to be funneled through the antenna complex that captured it initially to what's called the reaction center, where an important event takes place. This is the charge separation event. The electronic excitation is split into an electron and what's called a hole, basically a negative part, the electron part, and the positive part. And the electron-hole pair is completely separated so that the electron can now travel directionally down the electron transport chain to basically drive ATP production. And this is a similar electron transport chain to the one we find in respiratory complexes as well. So let's take a look at chlorophyll. On the screen here, you can see the Fenner-Matthews-Olsen complex. This is a well-known and well-studied photosynthetic complex. And, almost 100 years ago now, it was discovered that many chlorophyll molecules are required in a photosynthetic complex to produce just one oxygen molecule. And so this inspired the idea of collective light harvesting, a basic kind of elementary calculation by Emerson and Arnold showed that, in fact, thousands of pigments were necessary to collect enough photons in order to produce just one oxygen molecule during photosynthesis, which, of course, we know creates a sugar and oxygen. Basically providing both the food and the oxygen in the air we breathe to continue to allow respiration to occur for, as I mentioned before, all life as we know it on the planet Earth. So if you look inside the FMO complex, you can see there's quite a number of chlorophyll molecules, and these work collectively to capture light and drive those electronic excitations, the excitons, so to speak, to the reaction center. So, this Fennemathes-Olsen complex, for example, from green sulfur bacteria, serves as a crucial bridge between the antenna system and the reaction center itself, where the charge separation event occurs. So in this case, this well-known pigment protein complex enables tests of dynamic quantum theories. And in a famous paper by Engel and co-workers from 2007, they found that it appeared that a coherent wave-like energy transfer occurred in this system, which is very exciting. So we have this system with multiple chlorophyll sites. They have well-known energies and couplings. They've been studied very thoroughly. They're accessible via spectroscopy techniques, even in the ultra-fast regime. And as a result, they produce an ideal test bed to study coherent phenomena. As I mentioned, Engel and co-workers in 2007 identified coherent wave-like excitations in the system, and then this finding was reproduced at physiological temperatures in 2010. So in these ultra-fast spectroscopy experiments, lasers are used to pump oscillations to create these excitonic states, as I mentioned, both at cryogenic and even physiological temperatures. And the observation of wave-like dynamics between the chlorophyll pigments in these systems indicated that, in fact, there is delocalized coherent excitations. And this prompted the hypothesis that wave-like energy transfer may help to promote high light harvesting efficiency in photosynthetic systems like these. So these groundbreaking observations suggested that quantum coherence might play a key functional role in photosynthetic energy harvesting and transfer. Now, if we take a look at some of the data from 2010, for example, we can see the coherent oscillations, the rise and fall of the amplitude in the plot shown here, at different temperatures. So we see the most oscillations, the clearest at low temperature, 77 Kelvin. And as we go to higher temperatures, the oscillations begin to kind of fade away, but we can still see at 277 Kelvin, this is nearing physiological temperatures, that there's at least one, maybe two, if you squint about it, oscillations spanning out to 200 plus femtoseconds. Now, femtoseconds might seem a very short amount of time, but on the time scale of electrons, it's a long time. Long-lived coherent signals, upwards of 600 or even 1,000 femtoseconds or more, were discovered in the low temperature 77 Kelvin regime. And these, here's the problem, significantly exceed known excitonic coherence lifetimes, even at those very cold temperatures. And so it was later proposed that what looked like excitonic coherences must be something else. And in fact, it's now become, I'd say, fairly well established that these coherences are due to nuclear electronic, sometimes called vibronic, coupling and vibronic coherences. These vibronic states are hybrid states of matter that combine excited states of electrons with vibrational states of the nuclei. And this is another quantum effect, the fact that you can have not just a state of an electron or a state of the nuclear vibration, a phonon, but a hybrid quantum state that combines a superposition of both of these. So this, as the story continued, research in this area developed and the results were reproduced and theorized about theory of environment-assisted energy transport was proposed. The idea being that this phononic nuclear screening effect due to the molecular vibrations could protect the excitonic modes from decoherence and ultimately extend the coherence lifetimes. These hybrid states can then support directed energy flow to the reaction center, and the dissipative function of the phonons actually could help control the funneling of those excitations to the reaction center, reducing sensitivity to the surrounding molecular disorder. So now the picture of it is that in the FMO complex, interactions between these electronic states and the surrounding nuclear vibrations preserve the coherent dynamics from decoherence with respect to unscreened bare excitons, so to speak. These hybrid states are sometimes known as polarons because they basically reflect the polarized state of an exciton when it combines with phononic vibration. And what's interesting is that the dissipation is no longer purely wasteful, but it actually produces a useful effect of helping to channel energy robustly amidst diverse structural variations in these, organic biological systems, flexible biological systems, channeling the energy to the reaction center more effectively. So if we look at other model systems, for example, light harvesting complex 2, additional work by Arsenault and co-workers have shown that it also can exhibit vibronically assisted excitation energy transfer. If you take a look at the image on this slide, you can see light harvesting complex 2 is pretty big. It has an interesting kind of circular shape. And in additional studies in comparable systems also showed that the accurate reproduction of experimental spectra, when we do spectroscopy experiments on these systems, and accurate prediction of the kinetics, the reaction rates in these systems, demand realistic spectral densities. This basically tells you the density of different energies, different frequencies of vibrations in the environment. And what's critical to understand here is that simple exciton-only models, or models that include simple thermal noise interacting, with the excitons, fit some data, but can overlook crucial vibronic physics underlying energy transport. So the idea here is that, in fact, the thermal noise that was believed to destroy the coherence, actually, the presence of these phonon modes interacting with the exciton modes amounts to a quantum effect itself. Quantum decoherence doesn't necessarily just amount to, simple thermalization as the system relaxes, I would say naively, in our theoretical picture of it to a thermal state. But in fact, in this out of equilibrium system, just the interaction between the exciton and the surrounding vibrational modes can amount to a highly complex quantum effect that deserves study unto itself. And so you can read more about that in the references on this slide. Of course, in natural antenna complexes, the vibronic modes are very important because they are these dissipative modes that protect the system from, I wouldn't say overheating, but from being overcharged, from having the reaction center receive too many electrons, too many excitons to be separated at the same time. So vibronic kind of redirection is very important because the coupling between the exciton modes and the nuclear modes allows energy to be quenched by carotene molecules, the same molecules that make your carrots orange. These kind of quenching effects are critical in photosystem 2, for example, where energy-dependent quenching mechanisms prevent photodamage under bright light. And so there's a need for a kind of architectural balance in these photosynthetic scaffolds in order to precisely tune the system to optimize light harvesting, transport, and protection simultaneously. In low light levels, we want to harvest as much light as we can. And in high bright light levels, we want to funnel some, in fact, a lot of the excitons to the quenching sites so the reaction center doesn't get overloaded with energy. So how efficient is efficient? It's often been said, you may have heard of this yourself, that photosynthesis is incredibly efficient, nearly 100% efficient. And that is true. In low light conditions, these systems can achieve 95 or nearly 100% delivery efficiency as nearly all the excitons reach the reaction center and efficiently undergo charge separation, sending an electron down the electron transport chain to help make sugar and ultimately ATP, the energy currency of light. But in bright light conditions, the reaction center will saturate quickly, and this triggers protective mechanisms and structural reorganizations of the chromophores and the carotene pigments in the scaffold, in the light harvesting complex, and now the efficiency can drop very low. Total efficiencies based on the amount of energy absorbed with respect to all frequencies of sunlight can be as low as 5% in bright light. And so really what is kind of you want to consider as the true quantum yield, what I might call the refined photosynthetic quantum yield, is the number of successful charge separation events in the reaction center divided by the number of excitons that are delivered to the reaction center. It doesn't necessarily make sense to include the excitons that are intentionally quenched in our calculation because, of course, those losses are done on purpose in order to protect the system from being damaged to high light conditions. So the reaction center, nevertheless, has a yield of nearly 100% in both low and high lighting conditions, as every exciton that reaches the reaction center undergoes an efficient charge event that produces an electron to go down the electron transport chain without recombining or being lost, for example. So saturation and non-photochemical quenching are important processes to consider in these kind of systems. Now, if we only consider frequencies of light that are relevant to photosynthesis, then the saturation threshold has been suggested to occur at just 25% of full sunlight intensity. And so a naive estimate says that 75% of the energy must be safely dissipated to prevent damage. Now, there are other complex effects involved. As I mentioned on the previous slide, you also need to consider the percentage of sunlight that is even on frequency, on resonance with the photosynthetic reaction center. And an interesting characteristic of the carotenoids, the carotene molecules, is that even in low light conditions, now the carotenes can actually reverse their role of quenching, but help harvesting photons of some of these off-resonant colored photons that are not directly on resonant with the chlorophylls, but are sometimes absorbed efficiently enough by the carotenoids that the carotenes can pass the photon, the exciton, to the chlorophyll, which passes it to the reaction center. So you can now imagine an extraordinarily complex system that must use the network of carotenes and chlorophylls to efficiently capture as many photons as possible in low light conditions, and then to some extent reverse that process as the lighting conditions become more bright, now increasing in a very controlled way the number of excitons that are quenched. They're just thermally dissipated. So non-photochemical quenching acts as a controlled molecular switching mechanism between light harvesting and heat dissipation modes. And as far as we understand it today, it's likely triggered by a proton gradient formation across the chloroplast thylakoid membrane. And this kind of proton gradient across the membrane, of course, is reminiscent of the proton gradients that we see across membranes during respiration as well. And so in our study of quantum biology, we see many of the same kind of effects, many of the same kind of motifs in respiration as in photosynthesis, in electron transfer as in exciton transfer, and of course these effects are literally connected through the electron transport chain as well. Now, This is important not just for our understanding of biology or for our ability to genetically engineer or modify biology, but even for our development of artificial photosynthetic systems, artificial light capture systems. So we can learn lessons from nature, and in fact, an excellent amount of work, some excellent results have already been achieved. For example, the maximum fluorescence quantum yield has been optimized in carbon nanotube-based light harvesting systems to demonstrate that disorder and vibronic coupling, indeed, it's not just a theory, but can be used in practice to enhance certain areas of performance. And so already these kind of discoveries from quantum biology are being used to influence how we develop and ultimately the research directions that we take in developing hard technologies as well for light harvesting and other various areas of energy research. And 30% is a good number. This is simply where we are today, and as far as we know, research will continue to see the exact scope, the exact extent of the enhancements in these kind of systems that can be achieved through these kind of vibronic coupling mechanisms. So, is this really quantum? It certainly looks quantum. Electrons, excitons, proton mechanisms. People wonder how much of this really can be modeled classically. The underlying physics are obviously quantum mechanical, but if we can find a good model to model it classically, well then it's not exactly quantum computing, is it? So this is really the question as to what extent semi-classical modeling can be used to reproduce simulations of these systems, to reproduce the correct predictions, to accurately direct experiments. And so one particular study from 2022 asked the question of whether these kind of excitonic effects, energy transfer effects in photosynthetic systems are really quantum or classical. And they found that it's not exactly trivial, even in cases where a classical model will suffice. That if you want to take your truly dynamical quantum system and find a semi-classical approximation to match it, you still might need to do full quantum treatment of the system just to get your classical parameters for your model. Now, this again is highly non-trivial, and you might not want to do it by hand. In addition to your quantum mechanical computer simulations, you might want to use machine learning, AI-assisted parameter optimization, and pattern recognition in order to even figure out exactly what kind of classical model will best reproduce the underlying quantum dynamics. And in physics, we call this a coarse graining procedure, where we systematically reduce our very large scale, complex quantum mechanical description to an effective classical or semi-classical prescription for studying the system with these simplified model approximations. But don't be fooled. The classical model approximations must be derived from the correct quantum mechanical limits. And if you use a naive approach, just setting h bar to 0, for example, you can get very wrong-headed results. And naive classical models can even risk violating fundamental physics, violating Newton's laws, for example. Or even if they don't violate the laws of physics, they might just miss crucial couplings entirely if the thorough quantum mechanical analysis has not been done adequately in advance of parameterizing your classical model. So even in cases where classical models are sufficient to model quantum mechanical systems, quantum biology is necessary, I'd say absolutely crucial, in order to make sure that your classical model actually makes sense. There's not just one classical model that accounts for all quantum dynamics naturally. Each system may be different, and so the utmost care must be taken in translating fundamental quantum mechanical simulation into an effective classical model approximation. So do these quantum mechanisms confer an adaptive advantage? Well, certainly photosynthesis and electron transport during respiration are necessary for all organisms as we know them to live on Earth. So I would say that's an adaptive advantage. But people have wondered whether some quantum mechanical mechanisms have been harnessed by some organisms to achieve an advantage over other organisms. Is there a kind of quantum mechanical arms race or a technology race in biology to develop the best, most powerful quantum mechanical mechanisms at the molecular level? And there's some interesting research that has been done here to show that the Marcus inverted effect plays an important role in some, but not all, photosynthetic systems. The Marcus inverted effect is a true quantum mechanical effect with no classical counterpart. There is no analogous effect anywhere in classical chemistry for the Marcus inverted effect because it has the curious property in which the driving force, the free energy of reaction, as a chemist would say, as that driving force increases, the reaction rate slows down. And so it's as if you were driving a car and hitting the accelerator slowed down the car. There's really no classical way that that could happen. The accelerator is the accelerator, the brake is the brake. But in this kind of case, the Marcus inverted effect makes the accelerator act like a brake because of a curious quantum mechanical non-adiabatic effect. And so cyanobacterial photosystem 1 has been shown to exploit inverted kinetics in order to suppress wasteful back transfer. As the driving force goes up, the rate of electronic back transfer in the reaction center slows down, preventing wasteful back transfer and encouraging forward transfer of the electron after charge separation has occurred down the electron transfer chain. Vibronic coupling has also been shown to promote charge separation in a more general effect. This may be a general property of all photosynthetic reaction centers, where in order to achieve the directedness of the charge separation event and to ensure that it goes forward, not backwards, back to recombination, certain vibronic coupling effects create the necessary symmetry breaking effects to ensure directed charge separation down the electron transport chain and not back up it. And so organisms leverage these kind of tunneling and vibronic coupling effects to optimize photosynthetic coupling, photosynthetic function. Now, nuclear quantum effects are also often play a role in tunneling reactions, especially when the inverted Marcus effect is at play. Nuclear quantum effects are very common. They go hand in hand, you could almost say, with the Marcus inverted effect. And so these nuclear quantum effects are also likely to play a role in cases where the Marcus inverted effect controls the rate of electron transfer, not just in photosystem 1, as has been shown, but potentially in other systems as well. And so these all quantum effects all provide potential adaptive advantages beyond mere classical optimization effects. And these kind of effects do not have classical counterparts that can allow a simplistic classical theory of our system. They really are true quantum mechanical effects. Now, if we want to go beyond Marcus theory, well, and in fact, I would say sometimes we are forced to, because although many of our theories, Marcus theory and the various assumptions that go into electron exciton transfer theories, sometimes they work really well. Other times they begin to break down. So I've talked so far about the results that are well known, but there are anomalies. As in any field at the frontier of science, some of the results are well understood and other ones leave us scratching our heads. And so significant deviations from standard Marcus theory have been documented in electron transfer effects in photosynthetic system observed in purple bacteria reaction centers, for example, and these challenge our understanding of these processes. In some cases, there are uncertainty in how to calculate or what should be the correct electronic coupling matrix element beyond the usual Moser-Dutton ruler that is used in standard Marcus theory. Now, sometimes there can be kinetic coupling or gating effects, but in extreme cases, the Condon approximation, which actually dictates that the dipole coupling strength between the donor and the acceptor during electron transfer is conserved across the electron transfer process. Even this Condon approximation, which is generally taken for granted, can begin to break down. Now, as I mentioned before, antenna systems are also optimized to exploit disorder, and this kind of optimization defies our usual simple thermally activated picture of electron transfer, our transition state picture that ultimately still guides non-adiabatic electron transfer theory. And as a result, there are also non-trivial relaxation pathways. The system doesn't necessarily relax to equilibrium in just one way. And as is being shown, not just in photosynthetic systems, but in other light activated non-equilibrium systems, that the relaxation pathways can be critical, and the system's control of those relaxation pathways are essential in order to dictate the fundamental dynamics that drive specific processes. So I can recommend the references on this slide, particularly Krasilnikov, if you want to look into some of these kinetic anomalies. I think it's all very interesting, and there's still quite a bit of work that needs to be done beyond the usual approximations that we so often take for granted in the study of exciton electron transfer. So this means that photosynthesis is really a wonderful testbed for understanding open quantum systems. We have a paradigm, in fact, for studying open quantum systems dynamics, where we can consider the intrinsic vibrations of the system as the molecular motions shape the optical responses of the system and drive efficient exciton transport or robust noise or disorder resistant exciton transport through photosynthetic antenna complexes. These systems are optimally balanced for peak performance in what can be varying conditions, varying light conditions, varying temperature conditions, varying access to water. Is it the rainy season or is it a drought? The plant or photosynthetic organism has to adapt to all of these kind of environmental changes, and ultimately the structure and the interactions between the pigments are carefully adapted so that quantum coherences and the environmental dissipation that controls, even carves out through decoherence and dissipation, certain characteristics of those coherences, all must be balanced in order to give the organism, ideally, the best adaptive advantage it can take given the changing circumstances of its environment. So this immediately lends itself to the idea of design strategies. How does a living system protect useful coherences while at the same time harnessing structured environmental noise, even harnessing disorder in some cases, or at least creating robustness to disorder? And the question now is, what are the engineering principles that are needed to optimize the spectral density of the surrounding environment for optimum biological function? And just the few results I've shown and many more that you can read about in Chapter 4 of Physical Principles of Quantum Biology show how photosynthesis demonstrates how quantum systems can achieve optimal performance through dissipative engineering. So in this talk, we have considered photosynthesis as a quantum phenomena, and we have looked at effects like excitonic screening, which preserves exciton coherence vibrationally during photosynthetic light capture. We've looked at other vibronic interactions that enhance charge separation in the photosynthetic reaction center. We've considered how these kind of interactions can lead to robustness to this disorder and affects, for example, environmentally assisted quantum transport that help enable structure-independent transport, and ultimately help to produce the kind of directed effects that allow the exciton to reach the reaction center as needed, separate, and then go down the electron transfer chain with near 100% efficiency for the electrons that are meant to reach there prior to saturation of the photosynthetic system. Now, another interesting effect you can read about is the possibility of superabsorption, in which case environmental noise may enhance collective coupling, the collective absorption of light by multiple photosynthetic pigments in enhance effect, analogous to superradiance, if you have heard about this quantum mechanical effect, collective dissipative effect. And we finally discussed the Marcus inverted effect, which can prevent inefficient back transfer that would occur via charge recombination if the inverted effect itself did not prevent it by stopping the effective back transfer even as the driving force increases. So these kind of collective quantum effects can provide quantum enhancements to practically every area of photosynthetic energy capture, transport, and ultimately energy transduction, as that exciton is transformed into an electron going down the electron transport chain, which subsequently drives numerous biological mechanisms. Now, this all is very interesting because it presents the idea in quantum theory of dissipation not just as an unwanted source of noise, but as a quantum resource. And this has many broader implications for quantum technology because even beyond quantum biology, research in quantum information has shown that dissipation, in fact, can help with signal amplification, as noise and controlled loss can amplify some desired quantum signals. Ironically enough, dissipation can also enable fluctuation suppression, as environmental coupling reduces, in some cases, unwanted quantum fluctuations. Now, it's fascinating to think that dissipation can even be used for quantum state preparation, engineered to prepare strongly correlated states. And furthermore, that dissipation can be then used to stabilize these states, maintaining useful quantum states for quantum information processing, for example. And there's a number of recent, even very recent research in quantum information theory that shows that dissipation, really there's much more than meets the eye in terms of what kind of quantum mechanical processing can be achieved under highly dissipative systems. And this really opens the doorway to thinking about quantum biology very differently than it has been in the past, where living systems may harness dissipation to drive quantum mechanical effects that perhaps have not even yet been discovered. So, on that note, I will thank you for your attention. Please don't hesitate to download your guide to quantum biology, the physical principles of quantum biology, from the archive. It's available for researchers, students, and curious minds interested in venturing beyond the classical biology of last century and forging ahead to new vast horizons of quantum biology for this 21st century. I'm Dr. B. You can contact me at my email below, and don't forget to check out the rest of these lectures on YouTube. Thanks very much for your attention, and have a great day.